Okay, I'm David Pellman, and I'm very pleased to be able to welcome everybody to the Connect Science series today, and exceptionally pleased to be able to introduce our colleague from the Quadrangle, Lucas Farnog, who's the most recent young star recruit into the cell biology department. Lucas is originally from Germany, and after a brief high school flirtation with journalism, he has basically been on the express train for a really outstanding career in science and biophysics. He got his graduate education at the Ludwig Maximilians University, where he studied with Patrick Crema, and continued with Pat. That was for 4 years. And then had a brief stint as a junior group leader in the department in Patrick's department. Lucas has really changed the way we think about the eukaryotic transcriptome, and when he was in Patrick's lab, pioneered studies on how the RNA polymerase and chromatin remodelers interact with actual nucleosome chromatin. So please welcome Lucas, and we're looking forward to a very exciting presentation.
Yeah, hello everybody. Thank you very much, David, for the very kind introduction. I try to live up to the high expectations you just set with this very nice introduction. And I just also want to thank Claudia for taking care of all the logistics as well as the organization for the seminar. And today I'm really excited to share with you some novel biochemical as well as structural data that is really at the intersection of chromatin and transcription, looking at chromatin remodelers, histone chaperones, nucleosomes, and then of course also RNA polymerase itself, to really shed more light at the mechanistic basis of transcription through chromatin. But before I want to go into the nitty-gritty detail, I just want to point out how fascinating it is that a single cell can develop into a full-grown organism. And this, of course, requires intricate regulation of gene expression at all levels of the central dogma. And as David already introduced, I'm especially interested in the first step, transcription, where RNA polymerases transcribe the genomic information into RNA molecules. And I think we can all agree on that the central dogma is very beautiful in its simplicity. Of course, the devil is always in the details. And for that, let's just consider the DNA, for example. Eukaryotic DNA in the nucleus actually has to play quite conflicted roles. On one hand, the eukaryotic cells need to accommodate their large genomes in the tight confinements of the nucleus to protect it from detrimental effects. So compaction is required. But at the same time, the organism, of course, also needs to access its genetic information to replicate its genome, to transcribe RNA, and if it occurs, of course, also repair DNA damage. So these processes all require access to DNA. And as you can now imagine, accessibility, compaction, this is quite a dramatic logistical problem. So how are these interests of compaction and accessibility realized in eukaryotes? And luckily, evolution has actually come up with this ingenious protein nucleic acid complex that is called chromatin, and that we all know, that allows both for compaction as well as the controlled accessibility. And this becomes quite evident in this electron micrograph of this interface nucleus, where we see regions with high levels of compaction, here shown in this darker color, that we usually call heterochromatin, and then of course also regions of a lower local concentration of chromatin that we classically call euchromatin, shown here in this gray whitish color. But I think at the same time, it's really important to stress that fundamental nuclear processes such as DNA repair, transcription, as well as DNA replication really happen at nanometer scales. And the fundamental unit at this nanometer scale is, of course, not chromatin in its abstract form, but rather the nucleosome. And to just remind everybody what the nucleosome is, it's a protein nucleic acid complex that contains histone proteins, namely H2A, H2B, H3, and H4. And these come together for H2A, H2B in two dimers, and two copies of H3 and H4 together form a so-called H3H4 tetramer. And then all four histones, two copies of each of them, come together to form the histone octamer. And then 145 to 147 base pairs of DNA wrap around this protein core to form the classical nucleosome that we all know. And the nucleosome is, of course, the fundamental unit of chromatin. And I just want to use the opportunity now to introduce a little bit additional terminology that will be useful for the rest of my talk. The nucleosome has this quite peculiar property of having a pseudosymmetry axis. This pseudosymmetry axis allows us to rotate the nucleosome by 180 degrees, as you can see here in this animation, and we basically come back to the same superposition we had previously. And this pseudosymmetry axis around we just rotated, that is basically facing as the z-axis into the screen, is also known as the diet axis. And we can now exploit the diet axis to unambiguously define positions of the nucleosomal DNA on the histone octamer by first looking at the diet axis that is now shown here as this black line, and then calling the point where it crosses the nucleosomal DNA superhelical position 0. This is here at this position the case. And we can then count full helical DNA turns in a clockwise or counterclockwise fashion and name them unambiguously. In a clockwise fashion would be SHL -1, -2, and in a counterclockwise fashion we call them +1, +2, all the way to superhelical location +7. But what also becomes quite evident, I think, from the structure is the fact that the histone octamer interacts with the nucleosomal DNA quite extensively. There are usually 14 contacts that are being described in the literature. And the free energy of dissociation that would be required to unravel the nucleosome is estimated to be a whopping 12 to 14 kilocalories per mole of nucleosome. The nucleosome, and I think this is a very important property to keep in mind, is a formidable barrier which can be problematic for molecular machines such as Pol2. In fact, Pol2 will get stuck at certain positions within the nucleosome, and I will discuss this later in greater detail. But how do we then overcome this nucleosome that represents a molecular barrier to molecular machines? The cell employs chromatin remodelers, ATPases, to modulate the nucleosomal landscape. And generally, the activity of these chromatin remodelers is subdivided into three distinct categories. First, nucleosome assembly and spacing. Second, chromatin axis. And third, histone exchange. And we can use these different activities, as well as the subunit and domain composition, to subdivide chromatin remodelers into 4 different subfamilies, which are known as I-switch and CHD. And I will mostly be focusing today on the CHD family of chromatin remodelers. Then the switch-SNF family, which is important for basically removing nucleosomes from promoter regions to allow the transcription machinery to bind the promoter and start transcription. And in human, this would be the BAF complex as the most prominent member of the switch-SNF family. And we additionally have the 4th family, which is the ENO-A2 family, which is able to exchange certain histones for non-canonical histone variants. And 2 examples for that are, for example, exchange of H2A, canonical H2A for H2AZ in the early parts of genes, or the introduction of H2AX in response to DNA damage. But despite the differences in activities, all of these chromatin remodelers are actually molecular motors. They're ATPases that can translocate DNA. And this involves 2 lobes. And both of these lobes, which are called the Rec A-like lobe 1 and lobe 2, track along the same strand of DNA where one lobe, as you can see here in this animation from Janet Iwasa, sits slightly ahead of the other. And upon ATP hydrolysis, these motors can then move and facilitate translocation of the DNA. But as I already alluded to earlier, these chromatin remodelers have not only these two domains, the Rec-A-like lobes, but they also carry additional regulatory domains or form even multi-subunit complexes to move the nucleosomal barrier. So I specifically wanted to understand at a mechanistic level how these chromatin remodelers work. And the most approachable target at this time was the CHD family of remodelers, because they're involved in transcription, which is anyways the interest, my interest, but most of its members of the CHD family are also single subunit chromatin remodelers. And in the yeast Saccharomyces cerevisiae, there's actually only one member, which is named CHD1. And CHD1 is has a very modular domain architecture, again with this centrally located ATPase motor with lobe 1 and lobe 2, around which regulatory domains are arranged, namely a double chromodomain at the N terminus and a DNA binding region consisting of a SAND and a slide domain, here shown in this hot pink color, on the C terminus. So from a biophysical or structural biology perspective, it's really an ideal model system to study and try to understand chromatin remodeling. Specifically, I wanted to understand how first CHD1 engages the nucleosome, what functions the regulatory domains have, and then also learn more about the mechanism of DNA translocation that we have already seen in this previous animation from Janet Iwasa. But of course, we're not talking about two oven mittens that are moving on the DNA, but actual molecular machines. And we want to understand how this molecular mechanism works of DNA translocation. But because crystallization of nucleosome factor complexes is notoriously difficult, and we really only have, in the over 20 years of being able to obtain nucleosome structures, only obtain a handful with X-ray crystallography, I now switched over in 2017 to employing cryo-electron microscopy. And I was able to form a complex of nucleosome with CHD1 that was gel filtration stable, and collected a data set after sample preparation of 4,000 micrographs on a Titan Cryos microscope equipped with a K2 camera. And particle picking of the data then revealed approximately 1 million particles in the data, and subsequent to declassification revealed a nucleosome-like structure that you can see up here in these 8 panels, with additional density that were bound to it, that is CHD1. And using then 3D reconstruction methodologies, I was able to obtain the nucleosome CHD1 structure at an overall resolution of 4.6 angstrom. And using this reconstruction, we were able to build a model of this complex based on crystal structures of the nucleosome, the DNA binding region, as well as some additional de novo modeling that had to be done to obtain the model that you see now here. And what becomes obvious when we look at it is that CHD1 actually interacts quite extensively with DNA on one side of the nucleosome. Specifically, CHD1 binds with its ATPase motor at superhelical location +2, so 2 full helical turns away from the diet. Additionally, the double chromodomain, and we're rotating to this right now, as you can see here, contacts the nucleosomal DNA at superhelical location +1 via a basic patch interaction. But the most striking finding was that CHD1 actually is able to induce a non-canonical nucleosome conformation that was not previously observed, where the DNA binding region in this NCP conformation binds the opposite DNA gyre and seems to stabilize unwrapping of 2 full helical turns of nucleosomal DNA. And to visualize this, we have modeled here a canonical nucleosome with extra nucleosomal DNA, and we then morph to our structure, as you can see here. And I think you can appreciate the dramatic change the DNA undergoes quite easily. In fact, the DNA changes its trajectory by about 60 degrees. And this seems to really be a signature rearrangement of CHD1. And one can now, of course, based on this observation, speculate that this could potentially destabilize the nucleosome to allow for higher efficiency once the tightly regulated process of remodeling takes place. So this is how CHD1 engages the nucleosomal substrate. But we also wanted to understand the mechanisms of regulation. How does the cell make actually sure that the ATPase motor is not constantly hydrolyzing ATP and using up valuable energy? And to understand this question, a 10-year-old crystal structure from the Bowman lab at Johns Hopkins helped us actually tremendously. This crystal structure captured the ATPase motor with a double chromodomain in an inhibited state where the chromodomain sequesters lobe 2, and this we see here. It's the so-called chromo wedge that inserts into ATPase lobe 2 and prevents closure of the ATPase motor. So we're looking here at the inhibited state. We have modeled this onto our structure, and we now morph to the structural conformation that we have observed. And you can see now that upon binding of CHD1 to the nucleosome, the double chromodomain swings away from ATPase lobe 2 and interacts, as I described previously, with superhelical location +1, recognizing the bend of the nucleosomal DNA. And this swinging of the double chromodomain relieves the basic patch that was previously sequestered on ATPase lobe 2 and ultimately allows the ATPase motor with lobe 1 and lobe 2 to close and actively engage the nucleosome at superhelical location +2. It allows it to bind ATP to then start its remodeling activity. So this is an intramolecular regulatory mechanism how CHD1 can switch from an autoinhibited state through recognition of the nucleosome to an activated state that is fully engaged on the nucleosomal substrate. But regulation does not only happen intramolecularly, but also intermolecularly. As an additional regulatory mechanism to really control nucleosome recognition to make sure that the CHD1 ATPase motor is really engaging a nucleosome and not just a linear DNA, we were also able to observe interactions of the H4 tail with ATPase lobe 2, as you can see here in this transition. And a similar interaction was also observed for other chromatin remodelers, for example, SNF2 in the yeast system. And we think this probably describes a general mode of how chromatin remodelers are able to recognize the nucleosomal substrate. And overall, we think that this finding is especially important because H4K16 can be acetylated. And this is then one of the most important hallmarks of euchromatin. It is, of course, not difficult to imagine that the acetylation is also able to modulate interaction with CHD1, for example, by preventing binding into this cage here that we show in the red color. So this leaves us basically with the last question. How does the actual ATPase work? How do the molecular interactions between the lobes with its motifs and the DNA look like? And what you can see here in this depiction is that both lobes, lobe 1 and lobe 2, make really extensive contacts with the DNA via its various ATPase motifs. And based on structural data from an RNA helicase that is called VASA and another helicase that is called NS3, I was able to assign the tracking strand, which is defined as the strand on which all of these chromatin remodelers track along. And this indicates that CHD1 is a 3' to 5' translocase, which pushes the DNA once it does its cycle of ATPase hydrolysis towards the dye, towards superhelical location 0. So together with the great amount of biochemical work and structural data from other chromatin remodelers as well, we think we can now integrate and visualize the workings of this particular ATPase motor of CHD1, where ratcheting that is coupled to ATP hydrolysis explains the translocation movement, which ultimately provides the basis for chromatin remodeling. And in this state, the ATPase first binds DNA in a partially closed conformation. We see this here, and we call this the pre-translocated state. ATP binding then leads to complete closure of the motor and movement of lobe 2. And I'll switch to the next slide now. You will see how this closes. And ATP is bound in the active site of the remodeler. And this state we call the post-translocation state. And it also triggers DNA translocation by discrete 1 base pair steps. And then ATP hydrolysis would dissociate the ADP and resets the ATPase back into the pre-translocated state. And the enzymatic cycle can begin again. We have now visualized this in a movie again, where we go from the pre to the post and back to the pre-translocated state. So we're right now in the pre-translocated state. We now see binding of ATP and closure of ATPase loop 2 in this post-translocated state, and then subsequently reset upon ATP hydrolysis. And we go through these cycles more than once to basically start the chromatin remodeling activity and to shift nucleosomes on the linear DNA template. So taking everything together from the structural study, and by integrating this with additional available biochemical data, the nucleosome CHD1 complex really provides, I think, a good framework for understanding chromatin remodeling, especially because it's only a single polypeptide. Upon nucleosome binding, the double chromodomain releases the ATPase lobes, as I showed as this intramolecular regulatory mechanism. Cycles of ATP hydrolysis then lead to a movement of the ATPase motor in one direction, while shifting DNA towards the diet. This is shown here also in this little bulge. This pulls ultimately DNA in from the opposite side and makes the DNA longer on the proximal side where the DNA binding region of CHD1 binds, ultimately resulting in this shifting of nucleosomes and describing the activity of the CHD family of chromatin remodelers. So the yeast system was really ideal because it's just one family member in the CHD family. Whereas the CHD family has only one member in yeast, it has undergone quite a dramatic expansion in humans, where this family now has a total of 9 members. And these 9 members are categorized in 3 subfamilies, and they were able to acquire through evolution or even exchange certain domains. And this you can see here in this domain plot, essentially, of CHD1 all the way through CHD9. And I got particularly interested in the evolution of one member, CHD4, from subfamily 2, and how its domain architecture influenced its activity. CHD4 is an especially interesting target because it is implicated in heterochromatin formation and maintenance, which is, of course, significantly different from CHD1, which is known as a transcription initiation and elongation factor. And additionally, CHD4 is also part of multi-subunit complexes, which makes it, at least to our current knowledge, unique among the chromatin, among the CHD family of chromatin remodelers, where subfamily 3 and subfamily 1 seem to act more or less on their own. And ultimately, I was able to obtain the structure of a single copy and 2 copies of CHD4 bound to a nucleosome. And the single copy of CHD4 bound to the nucleosome is, as far as I know to date, still the highest resolution structure of any chromatin remodeler. And it shows overall, as you have probably already noticed, a very conserved architecture compared to the CHD1 chromatin remodeler in yeast. The most striking difference, though, is that CHD4 is not able to unravel nucleosomal DNA from the histone octamer, which could, of course, be consistent with its role in heterochromatin maintenance. But there was a minor inconsistency in my approach when I solved these structures, because I had used ADP beryllium fluoride to stabilize the ATPase motor of the yeast CHD1 on the nucleosome, and I used AMP-PNP for CHD4. And we were not 100% certain at the time if maybe also a difference in the ATP analog could result in a difference in the unraveling state of the DNA. So I established a FRET assay to monitor this process of unraveling and to verify if indeed CHD4 is responsible for the differences in the DNA trajectory, or if it is just rather due to the ATP analog. So this FRET assay that I'm employing here has 2 FRET probes on either side of the nucleosome. And we can then excite the donor and measure the emission from both the donor as well as the acceptor probe. as a proxy for how closely they are related, their distance from each other. We would then expect an increase in the donor emission and a decrease in the acceptor emission when CHD1 is bound, because the distances between the probes will ultimately increase, as we can see here, because we have this unraveling of the nucleosomal DNA. And indeed, this is the case for ADP beryllium fluoride being present together with CHD1, as well as for AMP PNP, and that you can see here on this plot, in this plot on the right-hand side, where we really see the increase in the donor and the decrease in the acceptor. However, for CHD4, the emission spectra does not change when compared to the control, giving, I think, very clear evidence now that CHD4 does not unravel the nucleosome independent of the ATP analog that is being bound in the active site of the chromatin remodeler. And I think, just to maybe extrapolating on a previous comment I made, this is then also consistent with the roles of these different CHD chromatin remodelers, where CHD1 again is a transcription initiation elongation factor that acts mostly in new chromatin, where CHD4 is implicated in heterochromatin maintenance, where of course unraveling and destabilization of nucleosome compaction are probably more unfavorable to really keep the nucleosome in a close conformation, and therefore you probably do not want unraveling of the nucleosomal DNA. So what I showed now was how chromatin remodelers can employ different auxiliary mechanisms of regulation to ultimately shape the chromatin landscape and to play different roles in different contexts, as demonstrated by CHD1 and CHD4. But the molecular machine that probably faces the most nucleosomes are actually RNA polymerases. And a rough back on the envelope calculation estimates that in a human, every second, approximately 50 to 100 nucleosomes have to be traversed by RNA polymerases. And in the nucleus, of course, this applies then to the 3 primary RNA polymerases. And over the past 20 years, they have actually been extensively studied and their structures have been determined. And as you might be aware, these 3 enzymes, of course, have a very similar core architecture, but perform very distinct roles. POL1 mostly transcribes ribosomal RNAs, and POL3 is responsible for the transcription of tRNAs as well as the 5S RNA, whereas POL2 transcribes most protein-coding genes in the form of mRNAs, as well as transcribing some snRNAs. And traditionally, of course, when we're now focusing on RNA polymerase II, what most of us learned in our undergraduate studies is that transcription of protein-coding genes is mostly controlled at the stage of initiation, where transcription factors recruit POL2 to gene promoters. But transcription, of course, is also regulated during transcription elongation, and this becomes more and more clear in the last 10, 15 years, where initially, after POL2 has escaped the promoter, capping enzymes will bind polymerase to cap the RNA, and then additionally, transcription elongation factors will bind POL2 to support its productive transcription through a gene. But something that has to be kept in mind the whole time is that the gene, of course, is decorated with nucleosomes that carry epigenetic information, and the nucleosomes are also there to maintain the integrity of the genome. One thing that the field has kind of not looked at all is chromatinized templates for structural studies. Interestingly, because of limitations in the structural methods, such as X-ray crystallography, there are over 140 structures of RNA polymerase II in the PDB, but we don't had, up until 2 or 3 years ago, any structure of POL2 on its native chromatinized template. And I thought this was a big gap, and I wanted to address this 3 years ago by providing the first structure of RNA polymerase bound to a mononucleosome. So the big question was, is it possible to visualize how POL2 transcribes a nucleosome, and then also, in a next step, start to understand how additional factors can support nucleosomal traversal by RNA polymerase II? And unfortunately, this is not completely trivial because it is simply not possible to just add RNA polymerase II and the nucleosome together and obtain a stable complex. They just don't bind each other if you just mix and pray, essentially. So I still wanted to tackle this problem to visualize for the first time RNA polymerase II on a nucleosomal substrate and open really a new frontier in our understanding of transcription in a chromatin context. So as a first approximation to address chromatin transcription, I designed a nucleosomal substrate that you can see here schematically depicted with a little bit of extranucleosomal DNA that allowed me to position Pol II in front of the nucleosome. And to my surprise, I was able in 2018 to obtain a cryo-EM structure of a RNA polymerase II nucleosome complex at around 4.5 angstrom resolution. And we see here that RNA polymerase II is in a post-translocated state. It sits in close proximity to the nucleosomal substrate, where the nucleosomal diatear superhelical location 0 faces towards the polymerase. And interestingly, the nucleosome seems to adopt a distinct conformation, which was completely unknown at the time. So again, we see here polymerase, downstream of it, the nucleosome, and this really defined interface between the polymerase and the nucleosome. But this defined interface was really surprising because based on bDNA and already available structures we had modeled previously, the approximate position of a nucleosome would have actually been rotated by a full 90 degrees. So we asked, what is going on here? Why is the nucleosome in a different position? And we were surprisingly able to see interactions of the nucleosome with Pol II. Pol II contacts the nucleosome at contact points, specifically, as you can see here in gray, the RPB1 clamp head, and in the sand color, the RPB2 lobe, that seem to interact with superhelical locations -1 and +1, and positioning and stabilizing the nucleosome in a certain conformation. And this finding was especially exciting at the time because the function of the clamp head and the RPB2 lobe were unknown. And additionally, both of these features of the RNA polymerase II in eukaryotes is not conserved in bacteria. So one can now speculate that these domains developed in step with the emergence of nucleosomes to really help the polymerase to orient nucleosomal substrates, chromatin, to facilitate transcription through chromatin. But of course, after achieving this first breakthrough of being able now to visualize Pol II and the nucleosome, the question was now if it is possible to extend the system to routinely study RNA polymerase II and its native chromatin template, essentially. Our first structure that I just showed, of course, hadn't answered many important questions regarding nucleosome transcription. We were not transcribing into the nucleosome in this thing, but just rather placing it in front of it. So now I designed a system that would allow the polymerase to transcribe into the nucleosome and then additionally employ transcription elongation factors that have been studied in the past, such as SPT45, and that always travel with polymerase during transcription elongation on this substrate with the bound nucleosome. Additionally, we also wanted to understand how histone chaperones, such as the dimer FACT, which consists of SPT16 and POP3, interact with the nucleosome in the context of the transcribing polymerase. And then also we were, obviously, very much interested in understanding how the chromatin remodeler CHD1 interacts with the polymerase, interacts with the nucleosome in context of the transcription, and then also, because this was known at the time, also understand how CHD1 interacts with the FACT complex. And one last question that we wanted to address here using the system is the in vitro loss of the H2A, H2B dimer. This is a conundrum that really has kept the field busy for the last 10, 20 years. It has been shown that Pol II on its own in vitro assays, when it transcribes a nucleosome, ejects the first H2A H2B dimer that it reaches. And when you lose this proximal H2A H2B dimer, actually Pol2 will further traverse into the nucleosome, and this will then result in a complete displacement of the remaining histone hexamer. But this is obviously not the case in vivo, where nucleosomes are even becoming more regularly spaced during transcription and stay fully intact without losing the H2A H2B dimer. And CHD1, in fact, this axis between the two plays an important role in stabilizing the H2A H2B dimer, preventing its loss and the loss of RNA polymerase II, but the loss of the nucleosome. But we do not have any structural information on how this could look like. So to answer these questions, I now extended the linear DNA that sits in front of the nucleosome, and then by adding NTPs to this reaction system, Pol2 can initiate transcription elongation and transcribe into the nucleosome. And we can now test how RNA polymerase transcribes through a nucleosome using classical RNA extension assays, as you can see here. And these RNA extension assays are based on the fact that we're using a fluorescently labeled RNA primer that can be extended by RNA polymerase upon addition of NTPs, as we can see here. And we do this in bulk, so many polymerases transcribe many substrates, and we can then, once we have accumulated a number of substrates, run these fluorescently labeled RNA substrates onto a urea gel that allows us to separate these produced RNA fragments based on their size. And this can then be used as a proxy to determine how far Pol2 has transcribed a certain RNA product, how much it has transcribed a chromatinized DNA template. And when we now look at such a transcription assay, first of all, what you can see is that we observe that Pol2 has the tendency to halt inside the nucleosome at very specific defined positions. For example, first, when the front of RNA polymerase II reaches the edge of the nucleosome, kind of what I showed in the structure that we solved initially, we can see that Pol2 starts pausing in front of the nucleosome. Then second, it is able to overcome this barrier eventually after enough time has passed. And we can see that when the active site of polymerase sits basically on the nucleosomal edge and the front of the polymerase touches the proximal H2A H2B dimer, there's again a mechanism for pausing involved during transcription elongation through a nucleosome. And then again, very notably, and this is the strongest barrier that we know of so far, is that when Pol2 is at base pair plus 42 and the front of the polymerase is close to the diet where the nucleosomal histone contacts are the strongest, again, we see a pausing effect here. And then ultimately, at least some polymerases are able to overcome these nucleosomal barriers and transcribe the full-length RNA product. And we can now use SPT45 as is shown here, like these, as is shown in these first 3 lanes here, as well as combinations of FACT, CHD1, and FACT and CHD1 to determine what effect these histone chaperones and chromatin remodelers have on the transcription efficiency to generate full-length RNA products. And what becomes super clear, I think, immediately is that FACT and CHD1 both have a stimulatory effect on nucleosomal traversal, which is generating up to 8 times fold more RNA product than does RNA polymerase alone with TF2S and SPT45. CHD1 on its own is maybe a little bit worse than FACT, but together FACT and CHD1 are again showing a very strong stimulation of full-length RNA product production. Interestingly, I also want to point out, and I'm not sure if you have noticed that, the presence of FACT also seems to slightly modulate actually the nucleosomal barrier at base pair 42, and this will become important in the later parts of my talk. So using these transcription assays, the big question that I had was now, is it possible to adapt these transcription assays and couple them to structural visualization via cryo-EM? And to do this, I again adapted my transcription assay nucleosomal substrate, But now, instead of feeding all 4 NTPs, I only use the subset CTP, UTP, and GTP. Additionally, my DNA sequence has an adenosine-less cassette up to 23 base pairs into the nucleosome, which means that for transcription in this cassette, only, as used here as well, CTP, GTP, and UTP are required. And this then enables POL2 to initially transcribe the first stretch of the DNA, but once it reaches base pair plus 23, where the first thymine is in the template strand, RNA polymerase II must stop because the complementary adenosine it would have to incorporate at that point is simply not available. And this trick now allows me for structural studies to at least control the heterogeneity of this reaction to some degree. But as you will see later, the sample is still quite heterogeneous and we have to deal with this as well. But because we have classification methods in cryo-EM where we can remove virtually heterogeneity that is introduced by the biochemical reaction, we can now, we for the first time have a fair shot at obtaining a structure of RNA polymerase II transcribing a nucleosome. And such an approach is really only possible with cryo-EM because in X-ray crystallography, we would never be able to crystallize essentially these heterogeneous samples. So to summarize this thought here, I use a sample preparation where I intentionally let a biochemical reaction happen at the order of minutes before I then further purify and freeze the sample. And I call this approach visual biochemistry. But of course, the question is, does visual biochemistry actually work? And the answer is yes, it does actually. I was able to form a complex with RNA polymerase II, the transcription elongation factor SPT45, CHD1, FACT, and the nucleosome, and then started transcription elongation by addition of the subset of NTPs that I mentioned previously. And through size exclusion chromatography, I'm then able to select A for actively transcribing RNA polymerase nucleosome complexes, and also check using SDS page analysis the presence of all my factors. So here you can see polymerase is there, SPT45 is present, as well as CHD1 and the 2 subunits POP3 and SPT16 of the histone chaperone FACT. And a denaturing gel that works very similar to the RNA extension assay that I showed previously, but just now run from one fraction from the size exclusion chromatography, also shows that I can enrich for a certain extended product up to base pair plus 23. But the sample is still a little bit heterogeneous, as you can judge here from the smear of bands that is present, as well as from the presence of RNA primer that is probably RNA polymerase that were not able to elongate the RNA. But nonetheless, I can take the relevant fractions, freeze them, and shuttle the sample into the microscope for imaging. And I collected an 80-hour data set on a Titan Cryos, this time equipped with a Gatan K3. And relatively quickly, 2D classes revealed RNA polymerase with densities that resemble the nucleosome that is here, seen from a so-called side view. And then additionally bound to that is CHD1, the chromatin remodeler, that you might have recognized now already from the earlier nucleosome CHD1 structure that I showed. And I was then able to, through extensive particle sorting as well as data processing that was also aided by machine learning, to reveal the structure of this POL2 SPT45 nucleosome CHD1 complex at a nominal resolution of 2.9 angstrom. And in this structure, POL2 has transcribed into the nucleosome until base pair plus 27. And this is, of course, very peculiar because the ALIS cassette that I used earlier and I introduced that was also used here actually was supposed to stop the polymerase at base pair plus 23. But when we look into the active site of the polymerase, we can be very confident that actually the polymerase has transcribed over the stop site because the resolution in the active site, as you can see here, is sufficiently high to call the purine or pyrimidine identity of individual bases, allowing us to assign the DNA register unambiguously. So we were doing here effectively sequencing by density features. And we think that this further transcription to base pair plus 27 is due to the fact that similarly to the nucleosome CHD1 structure, we also employed ADP rhodium fluoride here to stabilize CHD1 on the nucleosome. And we have now found out that ADP ordered from Sigma is actually contaminated with approximately 3% trace amounts of ATP. And these trace amounts of ATP that were present in the ADP beryllium fluoride preparation allows the polymerase to further transcribe 4 base pairs to this apparently stable intermediate. So what do we learn from this structure? We observe density for the transcription elongation factor SBT45 that is shown here in green that binds close to the upstream DNA, and the Cow5 domain of SPT5 that can be seen here binds close to the RNA exit channel. Additionally, RNA polymerase 2 has unraveled approximately 35 base pairs of nucleosomal DNA and seems to expose the first H2A H2B dimer. CHD1, in contrast, binds again very similarly to the structure observations from the nucleosome CHD1 structure at superhelical location +2. But notably, we do not observe any density for the DNA binding region of CHD1 that was, if you remember, bound to the opposite DNA gyre. And this becomes even more evident here in this comparison. Whereas the APO structure shows the DNA binding region binding the opposite DNA gyre, CHD1 in the pol2 structure that is shown here on the right side now does not show any binding of the DNA binding region, and we could also not visualize any density for it anywhere else on the DNA. And the reason for this is, of course, is because the opposite DNA gyre that is available in the APO structure is in the pol2 nucleosome structure inside the transcribing polymerase. Consequently, the DNA binding region can no longer bind in this transcribed state. And interestingly, there is actually older biochemical data that suggests that displacement of the DNA binding region can actually further activate the ATPase motor of CHD1. So what we're observing here is basically RNA polymerase 2 creating a nucleosomal environment through its transcription that further facilitates activation of CHD1 to basically make CHD1 help RNA polymerase 2 through additional chromatin remodeling. And to test if this hypothesis of chromatin remodeling during transcription is relevant and helps the process of transcription elongation, we designed a ATPase dead mutant of CHD1 here, D513N, and again performed a transcription assay, an RNA extension assay with this fluorescently labeled RNA. And indeed, the ATPase dead mutant of CHD1 is not able to stimulate nucleosomal traversal by pol2 as much compared to the wild type CHD1. And this is now the first time that one can demonstrate in a minimal biochemically reconstituted system that chromatin remodelers are not only chromatin remodelers, but that they're also stimulatory transcription elongation factors. But there's one question I couldn't answer yet, and this is the question why we not observe FACT. FACT was actually present in the preparation. I could already, and I did show you in the early SDS page gel that also both of these subunits were in the complex, but we did not observe any density for the FACT complex. And to address this, we now decided to just form a similar pol2 nucleosome complex, but leave out CHD1 to only use FACT with the hypothesis that potentially FACT and CHD1 share a surface, and CHD1 just would displace FACT on the nucleosomal substrate. And by forming this pol2 SPT45 FACT nucleosome only complex, we were able to obtain a structure at 3.1 angstrom resolution. Again, SPT45 is bound at with its NGN domain, it's bound at the upstream DNA, and the Cow5 is at the RNA exit channel. And notably, instead of transcribing to the designed positive base pair +23, also in this structure, RNA polymerase 2 has only transcribed to base pair +17. So it's basically stalled 10 base pairs before the corresponding CHD1 complex. But nevertheless, the polymerase is able to position the nucleosome in a very similar orientation observed to the other two Pol2 nucleosome structures. So what we think is happening here is that there are certain orientations of nucleosomes when polymerase has transcribed to different positions that are so energetically favorable for Pol2 to adopt together with the polymerase that Pol2 only pauses, has barriers when the nucleosome is not rotated in these particular ways. So what this means is polymerase pauses potentially at positions that are energetically favorable when transcribing through a nucleosome. And these are the positions that we observed previously on our transcription assay. At the same time, it might also be possible that these specific orientations are adopted to also recruit or couple the recruitment of other factors to this orientation to be a binding platform to then help RNA polymerase II or epigenetic modification to happen co-transcriptionally. So with this structure, let's additionally have a closer look at FACT to try to understand this histone chaperone and its histone chaperone activity. FACT is now bound to the partially unraveled nucleosome again that I showed you. It's this time just 25 to 30 base pairs that are unraveled. And with the two subunits, FACT binds across the diet of the nucleosome with the two subunits, kind of forming a saddle that sits across the unicycle that is being formed by the nucleosome. And when we now use these structures and superpose them, we can clarify why we could never observe FACT when CHD1 was bound. I have created here a superposition of the FACT and CHD1 structures viewed from two orientations. And it is pretty clear immediately that the binding surfaces of CHD1 and FACT overlap on the nucleosomal substrate. The double chromodomain of CHD1 and parts of FACT, specifically POP3, clash with each other. So only one of these two factors can bind at a time. And this is why we think we only observe CHD1 in the Pol2 CHD1 nucleosome structure. It's just binding stabilized to ADP beryllium fluoride much more stably to the nucleosomal substrate, and thereby excludes FACT. But you might have mentioned that I didn't mention one additional structural observation that we made, and this finding concerns the C-terminal domain or CTD of SPT16. As expected, transcription again has resulted in this partially unraveled nucleosome where the proximal H2A and H2B dimer is exposed. And the CTD of SPT16 now takes the place of the unraveled nucleosome and stabilizes the H2A, H2B dimer to prevent it from dissociating from the nucleosome. And this is a super critical feature of the FACT complex because biochemical data from the Reinberg and Luger lab had already shown about 7 to 8 years ago that loss of the proximal H2A, H2B dimer results in complete nucleosome disassembly during transcription. And of course, the cell needs to prevent this from happening because if we lose the nucleosome, we also lose the epigenetic information that is present through histone marks on this nucleosome. So ultimately, the cell would lose critical information about the transcriptional state of a gene if every RNA polymerase II completely ejected nucleosomes that is transcribing through. So FACT simply through binding energies stabilizes the H2A, H2B dimer by mimicking the unraveled DNA. It maintains the nucleosomal landscape and facilitates transcription through the nucleosome. So taking both structures, we end up with a model now for factor-mediated nucleosome transcription, where transcription into the nucleosome bound by CHD1 results in the displacement of the DNA binding region of CHD1 and the activation of the ATPase motor. CHD1 can, through this, support nucleosomal transcription until exposure of the proximal H2A, H2B dimer. And FACT can then eventually displace CHD1 to bind the nucleosome to stabilize the exposed proximal H2A, H2B dimer. Pol2 can further transcribe, traverse the nucleosome, and prevents loss of the epigenetic information that is stored in the nucleosome. So the important insight really from these structures is that the Pol2 transcription machinery and Pol2 transcription into the nucleosome generates a nucleosomal environment that allows A for the binding and B for the activation of factors that support transcription. And these factors that are binding in these contexts are specifically equipped to recognize these non-canonical nucleosome conformations that are being generated during transcription. So to put this all in a nutshell, integrated structural biology can be utilized to understand basic mechanisms of nuclear biology at the intersection of chromatin and transcription and help us to understand the regulation of gene expression. And with this, I just want to quickly use the occasion to thank my former mentor, Patrick, who supported all of this work, and also thank my current lab members, Yeli, Allison, and Martin, who have started in the last few months in my lab to start working on these problems of chromatin transcription. And I thank you very much for your attention.
That was a spectacular talk. I'll start with some applause. Just for the audience, I'd like to ask that you put your questions in the Q&A just to remind you and not raise your hand. So I'll pull this up. So Kwang Yi is writing, thanks, Dr. Farnog, appreciate your sharing as well as thanks, was wondering whether you would be able to share, one, the binding affinity for CHD1 domain to ATP, and two, the proportionality of the conservation of binding affinity energy to atomic force. I hope my question makes sense.
Yeah, absolutely. So I don't recall the exact binding affinity for ATP to CHD1 right now, but I can just point to work from Greg Bowman, who has very carefully looked at this problem. And what is very interesting, the transition state analog ADP beryllium fluoride, when bound to the CHD motor, actually has a half-life of greater than 24 hours, which is really incredible. ATP, of course, the assumption would be it binds into the active site and then gets ultimately immediately hydrolyzed, and then it would dissociate again. And then towards the second question about the proportionality of the conversion of binding affinity energy to atomic force and so on, this is something that is, of course, still very interesting right now to look at. And I think there are people who are interested in single molecule studies that are trying to figure out exactly how every ATP hydrolysis cycle is actually coupled to the chromatin remodeling event, so the shifting by one additional base pair. But this right now is still a matter, I would say, of investigation, essentially, how effective these chromatin remodelers are in terms of ATP hydrolysis to chromatin remodeling coupling.
Juntao Yu says, great talk. Lucas, I'm wondering whether if you change the location of the ALIS cassette on the DNA template, will it change the location of the back CHD1 Pol2 in the structure? Thanks.
Yeah, this is something that I've been thinking a lot about, because one of the problems, of course, is that we know that CHD1 especially binds at the superhelical location +2, and I would assume that it in all contexts binds there. But of course, when the polymerase, for example, has only transcribed a few base pairs into the nucleosome, or when it has transcribed over the diet, suddenly there is actually no space for the CHD remodeler to bind. So I would say yes, in the context of positioning the ALIS cassette at different spots, we can probably modulate at least if CHD1 is bound or is not bound. For a fact, we know actually through comparison with data from Caroline Luger that FACT can obtain or adopt different conformations on the histone octamer surface. In our case, the structure that we have is shifted by one full superhelical location compared to Caroline's structure on the subnucleosomal substrate. So I would assume that by modulating the presence of the ALIS cassette or the position of the ALIS cassette, we can probably also modulate, at least to some degree, where FACT actually binds.
Okay, well, I'll take the liberty to ask one question myself. There's recently been a lot of interest in the possibility that nucleosomes can undergo a phase transition and that this is contributing to the establishment of different compartments. And I wondered whether this is something you considered or have any speculation about how it would alter these highly dynamic interactions that you've revealed.
Yeah, I think this is like one of these next steps to go really from like a, I would say, rather nanometer look to maybe get a little bit higher macrometer, so to speak, like to go to a little bit more macromolecular level to look at that. I think it will right now be difficult to reconstitute this, at least from the ground up in my system. But I think cryo-electron tomography, for example, will be a great tool going forward in the future, because we can, FIB cells by now. And then I think also with the structures that we have solved now, we can kind of start template matching certain features inside of the nucleus and try to understand phase-separated entities in there. And I think this is additionally the great advantage that we can maybe also couple this tomography with fluorescent light microscopy-based methods and really then also check what is causing these phase-separating domains, so to speak. Is this epigenetic modifications such as H4K16 acetylation? Is this the presence of certain factors that also help in this context? So I think it's very exciting to do this, right? But I guess we'll have to work on that.
Wonderful. Looks like this is the end of the questions. I'm sure if questions come to people later on, feel free to contact Lucas. I'm sure he'd be happy to address any questions that come up. And again, Lucas, I just want to thank you again for a spectacular talk and a great sort of view over the horizon at where the future of the transcription field is going. Thank you.
Yeah, thank you very much, David.